Welcome to all. Behalf of Homeopathic Research and Development Organization, once again welcomes. So we are conducting a series of webinars about homeopathic research and development and few awareness videos about homeopathy to the peoples. Today we will discuss about the essential pathophysiology of SARS-COVID for doctors who are in the field of homeopathic practice. This is Dr. Suresh, a faculty from Homeopathic Research and Development Organization. So all the work are dedicated to our homeopathic master, Dr. Samuel Hahnemann, and to my eye opener, Dr. Praful Vijayakar. Outline of the session. Today, what we are going to see is first the knowledge of disease. Based on the aphorism number 3, we have to acquire the knowledge of disease and knowledge of homeopathic applications. So in the first video, we will see about the knowledge of disease, about the medicinal part. So about the coronavirus first, then pathophysiology when enters in the human body, morphological finding, clinical features, and investigation. These are all the practice of medicine part. Then in the next video, we will see about the homeopathic approach. First, we'll start with the history of corona from history. Coronavirus were first discovered in 1930s when an acute respiratory infection of domesticated chickens. Human coronavirus were discovered in 1960s. In 1965, successfully cultivated the novel virus and found lipid envelopes in the coronavirus. Other human coronavirus have since been identified, including SARS virus at 2003, H-COVID NL63 in 2004, H-Co Hong Kong virus at 2005, MERS coronavirus at 2012, and now it is a SARS coronavirus second generation at 2019. There have been large number of animal coronavirus identified since 1960s. So these are all the history about the coronavirus. Now we'll see about the present pandemic SARS coronavirus 2019. Coronavirus 2019 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. Severe acute respiratory syndrome coronavirus 2 is a novel coronavirus first isolated from the 3 people with pneumonia connected to the cluster of acute respiratory illness cases in Wuhan state of China. In February 2020, the Chinese researchers found that there is only one amino acid difference in the binding domain of S protein between coronavirus from pangolins and those from humans. However, whole genome comparison to date found in most 92% of genetic material was shared between pangolin coronavirus and SARS-CoV, so which is insufficient to prove pangolins to be the intermediate host. It is nothing but in 2020 the Chinese researchers found that there is only one protein molecule is differs from coronavirus of human beings as well as coronavirus of pangolins. Pangolins is nothing but it is a kind of animal. So we can common name we can call it as ant eater. So the human gene of coronavirus and animal pangolin source of coronavirus are 92% similar, though It is a insufficient evidence to prove the pangolins are the intermediate host of coronavirus. Then we'll see about the structure of the coronavirus. Coronavirus is large, roughly spherical particles with bulbous surface projections. Right? This bulbous surface projection. These are the bulbous surface projections. The viral envelopes consist of lipid layer, then membrane layer, spike proteins. So these pink color structure are the spike proteins. Then this red color are the envelopes. Then okay, coronavirus contains a positive sense signals. So from these spike proteins, coronavirus are sending the signals. So this is the simple morphological structure of the coronavirus. Now we'll see the pathophysiology. How the coronavirus enters the human beings, how it replicates, and how it's producing symptoms in the human beings. This is collectively called as pathophysiology here. So we all know the lungs are the organs the most affected by the COVID-19. In the lung alveoli are the in the lung alveoli there is the two types of cells. One is pneumocyte type 1 cells and pneumocyte type 2 cells. Pneumocyte type 1 cells is responsible for gases exchange in the respiratory membrane. Second one is pneumocyte type 2 cells producing surfactant to the alveoli. The virus access host cell via the enzyme called angiotensin converting enzymes 2. That is short form is called as ACE2. These ACE2 are the receptors which is binding the coronavirus into the human cells. The density of ACE2 in each tissue correlates with the severity of the disease. So whenever the ACE2 cells, ACE2 inhibitors are more, the coronavirus infections also possibility is more. So decreasing the activity of angiotensin converting enzyme 2 can leads to be more protection against COVID virus. So what happened? This ACE, so here, this is then ACE antibody. This what happened? This connects the COVID virus as well as the pneumocytes. This is the one material which links between two things. Then how pneumotype 2 pneumocyte type 2 has ACE2 receptors. The coronavirus spike of glycoprotein fits into the ACE2 receptors. See here, these are all the some kind of on angulation kind of structure is there. No, this structure are the ACE2. This connects the coronavirus to the pneumocyte type 2 cells. So this is the linkage. Now what happened? As soon as virus connects to the pneumocyte type 2 cells, it engulfs and phagocytosis starts. During the phagocytosis, lysosome digests the lipid wall of the virus. So what happened? As soon as engulf the virus, the lysosomes in the cell try to act over the virus. Due to the action of the lysosome, the cell wall, the protein structure, the lipid structure of the cell wall is digested. Then the RNA of the virus exposed directly to the human cells. Now the RNA of the virus enters into the cytoplasm, turns as the messenger RNA. So this spiral RNA now becomes the messenger RNA. There it transcript into many RNA due to the polymerase reactions. So after entering in the mRNA, it transcript into the many RNAs, right? It's a copying. So this is how the RNAs are forming. It's RNAs are the inner material of the virus. So the inner materials are formed. Now what happened? There is another cell structure named as a ribosome. Ribosome translates and form different types of polyprotein at the same time. So where the messenger RNA it transcript into the many RNAs at the same time, this is their activity from lysosomes. Now the ribosomes acts, liposomes secretes many polyproteins to repair the cell injuries. But beyond that certain limit, what happened? The polyproteins are clumped and collectively forms capsule of the spike of the virus. So this is how the outer wall, outer covering of the virus are formed. Now already the inner structure of the virus are formed from lysosomal activity. Now due to the ribosomal activity, the capsules are formed. Now both capsids and RNAs both are collectively joined and forms new virus. This is how the replication of new virus happening inside the pneumocyte type 2 cells. These RNAs are combined with the fragmented polyprotein capsid and spikes generated by the ribosome, resulting in forming of new coronavirus inside the cytoplasm. Right? I think it's a simple thing. You can understand it simply. Need not to be thing. Even you can read the PPT also. You can understand the things which comes out from the pneumocyte type 2 cells. Now what happened? The new virus are formed. It breaks the pneumocyte type 2 and comes out and try to infect the neighboring cells. So this is how diseases are progressing in the lung. So what happened? So this is how the new virus are forming. New virus are forming. So this is the RNA structure. This is the capsule combines and joins. New virus are forming. Now it breaks the cell wall and comes out of the cell wall. This is on simple illustrations. Now what happened after during this activation during this active process of replication of RNA virus. Now the morphology, the macrophages. Macrophages are the one of our soldiers in our immune system. When the infected cell burst, so this is pneumocyte cells. Pneumocyte type 2 cells are burst. They releases some inflammatory mediators, so it's sending some signals to the immune system, because I have a problem, I'm suffered. You have come and survey my neighboring friends and neighboring colleagues. This is what the signal sending from the infected cells. So that signals are nothing but the inflammatory mediators. The inflammatory mediators like interleukin 1, interleukin 6, interleukin 7, tumor necrosing factor alpha, and few others. These are all sent to the macrophages. These mediators enters from alveolar space into the blood and act as a vasodilators. So we might all aware of that inflammation. During the inflammation, we studied that the vasodilatation is the first process of inflammation, the first step of inflammation. So when these signals are enters into the bloodstream, immediately the blood vessels are become dilatation. It's called as vasodilatation. So vasodilatation occurs, vasodilatation of alveolar capillaries, and so once the vasodilatations occur, the permeability are increased. So when the permeabilities are increased, what happen? See, the vessel wall permeability is increased. The plasma accumulated into the interstitial space. Then the plasma accumulates into the interstitial space. Then compression of the alveolar and some other fluids enter into the so so this is the vessel wall. So due to the vessel wall permeability, this fluid from the vessel goes to the interstitial space. So this is the space between capillaries and alveolar wall. This is called interstitial space. Here lot of fluids accumulates, causing this fluid accumulation can causes alveolar edema. So due to the alveolar edema, the surfactant of the alveolar are sinking in the things. This is for a simple understanding. This is the cut section of the respiratory membrane, that is the alveolar wall, how the gases exchange is happening. So this is from alveolar side, this is from capillary side. This lined area is there, no, there's a spiral or some lines are there, no, this is the interstitial space. So first innermost blue line is called as surfactant, then alveolar epithelium, then basement membrane of the epithelium, then interstitial space, then come to another side. This is the basement membrane of the vessel. This is the vascular capillary endothelium. So this is how the gases exchange are happening. So from alveolar side, oxygens are goes this side, carbon dioxide goes this side. This is how gas exchanges are happening. Due to the increased surface tension, the alveoli collapses. So when the lot of fluid accumulations are there in the interstitial space and the surface tension of the alveoli is collapsed, the accumulation of fluid between the capillaries and alveolar interrupt the gases exchange. This is how the hypoxia starts in the COVID cases, severe cases. Now, at the same time, during an inflammatory reactions, many neutrophils enter. Now, after the macrophages, now neutrophils enters into the Warfield, which releases reactive oxygen species and protease. So, after the macrophages, some neutrophils also enter into the action. So, due to the its release of the oxygen species and proteases, what happened? These proteases can destroy the virus. So when destroying the virus, it destroying some parts of the cells also, that is type 1 pneumocytes. Still no problem with only type 2 pneumocytes. Now only type 1 pneumocytes are destroying. Type 1 pneumocytes are responsible for gases exchange. So it leads to further worsening of hypoxia. The destroyed cell along the some protein collects in the alveolar cavity. So now the fluid accumulation, few cells are destroyed and some debris are forming. This is known as consolidation. Then it leads to septicemia and septic shock due to increased permeability and edema. So due to the septic shock, blood volume and hypotension. Now perfusion also reduced it to the other organs, so can lead to multi-system organ failure. If the inflammation of the lung spreads into the bloodstream, become systemic inflammatory response syndrome. So till now the inflammation and the problems is only in the respiratory system and respiratory membrane. Now what happened? This viremia, due to this viral overload, the inflammatory response, the cytokinin activities are spreading to the other systems via the blood streams. Now the perfusion of the oxygen supply to the other organs also reducing, can leads to multi-system organ failure or systemic inflammatory response syndrome. Same time, inflammatory mediators like interleukin 1, 6, reaches the hypothalamus. So in the bloodstream, it reaches to the hypothalamus, stimulates the prostaglandins, it stimulates the prostaglandins, which causes increased body temperature. That's called as fever. Same time, so due to the compensatory mechanism, heart rate is increasing and respiratory rate is increasing to manage the hypoxia by stimulating the sympathetic nervous system. Then low blood pressure and anasarca follows. So if the perfusion of kidney gets less, causes elevation of blood urea nitrogen creatine. If the blood flow to the liver leads to elevation of the liver enzymes and bilirubin, then collectively kidney, lung, kidney, liver, so one by one organs are suffering, then can lead to multi-system organ failure. So there is another condition, coagulopathy, disseminated intravascular coagulation. Many of them aware that the apart from pneumonia, the peoples are dying, peoples are suffering. Some other small vessel blood coagulation or small vessel blood thrombosis. So this is nothing but it's called as disseminated intravascular coagulation due to the overactivity of inflammatory mediators like interleukin 1, interleukin 6 and TNF can trigger the acute endothelial cell activations of the small vessels. So endothelial cells lost normal homeostasis. So due to this inflammatory mediators, triggering the inflammatory mediators, the endothelium of the small vessels lost its normal mechanism, normal functions. So what happened? Which regulates the fibrinolysis. Now it loses the uh, it loses its activity. Now vessel wall permeability and local endothelial cell dysfunctions in in the pulmonary microvasculature. Now the thromboinflammatory process in COVID-19, vasculopathy, ventilation perfusion mismatch and the clinical phenotype of refractory acute respiratory distress syndrome. This is how happening. Simply, these inflammatory mediators can cause the endothelial cell, a kind of endothelial cell injury in the small vessels. It loses its the functions. Now, so when the injury happening inside the vessel walls, they try to form the thing, try to seal the things. This process is called as coagulopathy, disseminated intravascular coagulation. This is one of the commonest cause of the death in COVID-19. Now, for our convenience, we can convert this, we can segregate the stages as a three stage. One is upper respiratory infection from nose to throat. Second one is from throat to bronchus. Then bronchioles. Then alveolar and multi-organ involvement as considered as the stage 3 for our convenience of our study. Now we'll see what is the pathological findings in stage 1, stage by stage. First, stage 1. So stage 1 is from nose to throat. So usually the nasal mucosa is erythema and edema are common, congested and inflammation of the pharynx and tonsils. This is the stage 1. Then stage 2. During the episodes of acute bronchitis, mucous membrane hyperemic and edematous, diminishing of bronchial mucociliary functions, the air passage clogged by debris and irritations increases, copious secretions of mucous membranes. Then stage 3. This is histologically COVID-19 shows the diffuse alveolar damage corresponding to the phase of disease. This is what we have seen still now in the stage 3. We studied most about the stage 3. Then leads to systemic inflammatory response. Then perfusion to vital organs are reduced. Then features of sepsis and multi-organ failure. This is the stage 3. Then we'll go to the, so whenever we study the diseases, starting from definition, then prevalence and histories, then pathophysiology, then morphology, then clinical features, then management. This is the structure we followed from the college days. So the same way I'm presenting this. Then this is the clinical features. Clinical features of stage 1. Stage 1 is simply the upper respiratory infection. The symptoms of upper respiratory tract infections like other flu or other seasonal cold, very similar things. It might be the self-limiting cold, coryza, throat pain, fever, cough and catarrh, profuse watery nasal discharge. Then leads to sometimes there is a possibilities of cervical lymph node enlargement, conjunctivitis, hoarseness of throat, throat pain, headache. These are all the features of upper respiratory tract infection. Then the stage 2. The stage 2 is from throat to bronchioles. Now the windpipe and bronchi both carry air to the lungs. So when they become inflamed, it usually results in some difficulty in breathing, right? From throat to bronchioles, it goes no. So once there is some inflammation, some irritation, what happens? It loses its homeostasis. That is, it loses its physiological function. So its main function is carry the oxygen to the lung. It affects. So it causes difficulty in breathing and severe cough. Maximum of dry cough is possible. Fatigue, wheezing, nausea, vomiting. Nausea, vomiting are due to the stimulation of the vagus. Then stage 3. This is a highly complicated stage. It's one of the needs some serious medicinal intervention. So it is a dyspnea, shortness of breath, cough of the productive mucus. It is almost a sign of pneumonia, sepsis, multi-system organ failure. This is almost, we can call it as a terminal stage. It's very difficult to revert in this stage. Increased heart rate, increased respiratory rate, hypoxia due to stimulation of sympathetic nervous system, and a low blood pressure. So these are the clinical features of 3 stages. Then we'll go for the diagnosis and investigations. How to diagnose the case? What are the investigatory methods? So we can for our convenience, we can take there is a 2 type investigation. We can divide into 2 types. One is blood investigations. Second one is imaging. Blood investigations are nothing but things. Blood investigations. I am included in the swab test also in the blood investigation, because it's not it's not comes under imaging. Nasal swab or pharyngeal swab through RT-PCR. And another test is NAT. That is practically that is not doing nowadays, the NAT, because it's highly it's very costly and a costly procedure. So nobody is preferring this NAT. Then CBC finding as lymphocytopenia. So it's an acute infection, acute stage. So in the acute stage, we can find the neutrophil surge and the macrophages. So automatically chronic inflammatory response like lymphocytes activities are reducing. Now, and the liver function test can causes increased ALT, AST, bilirubin. The renal function test, when you assess the renal function test, blood urea nitrogen and creatines are increased because the perfusion to the kidney is reducing, so it suffers. Another thing is procalcitonin. If the secondary infection with bacteria or something, we can find the procalcitonin in the renal function test. Other general markers like C-reactive proteins, erythrocytes, ESR elevation, interleukin, then D-dimer. D-dimer is a very important factor in case of COVID-19, because D-dimer is assess the disseminated intravascular coagulation and internal bleedings. Increased ferritin level also possible. Elevated troponin I and creatine kinase thing for them to find the activity of the heart. Then imaging. There is chest X-ray. Chest X-ray we can get commonly ground glass opacity. It's very difficult to distinguish with other things. Then CT chest. CT chest is very important investigations in case of stage 3 COVID infections. So there you can get ground glass opacity in the peripheral lung field, because peripheral lung field is composed of mostly the alveoli. So the stage 3 or most of the COVID cases, alveolar involvement is more. So only the peripheral ground glass opacity, the consolidations also possible. So I already explained about how the consolidations are forming. Then crazy paving pattern, interstitial thickening. This I will show you the pattern in the next slide. Then another imaging technique is USG chest, pleural line thickening, increased B lines, consolidations with air bronchograms. I will show you the images in the next. So this is of crazy paving pattern in the CT chest. So this is the peripheral field. Here this is crazy paving patterns, and this is called as B lines in the USG. B lines, right? So thank you for all your cooperations. So in the part 2 we will see the homeopathic management, etc. Thank you.